In this video, we introduce Thrive, the Tumor Heterogeneity Research Interactive Visualization Environment, and demonstrate some of the current functionality of our software. We start by selecting the tumor sample and fluorescent marker that we would like to visualize. In this case, we choose the DAPI channel of a specific colon cancer spot in one of several TMA slides in order to observe the cell nuclei. Any imaging platform which outputs TIFF or JPEG files can be used to generate data for our software. Thrive allows us to mix and match among cell segmentation, quantitation, and heterogeneity algorithms. In this case, we demonstrate one example of each, but more of each will be added to future software updates. Here we choose the wavelet nuclear segmentation algorithm. We can run the segmentation after simply setting some default parameters such as minimum and maximum detection size. Otherwise, this method is completely automated and does not need a user-defined segmentation threshold. We type a name for the result set and then scroll down to see the list of algorithm executions and execute the algorithm, which takes about 10 seconds, on par with typical running times for similar methods. We see that the algorithm completed successfully. We click to view the results. The cell segmentation mask can be inspected and its opacity interactively adjusted to visualize the underlying fluorescence image. This is one straightforward segmentation algorithm. Thrive will provide several others currently in development and also an SDK for the research community to contribute still others. Thrive is intended to be a platform by which the cancer research community can try, evaluate, and compare algorithms. A simple form allows the user to provide feedback, and aggregated comments over time help rank alternative algorithms. In the future, we plan to solicit more detailed algorithm feedback, even annotations, to improve the algorithms themselves. Next, we run an algorithm to quantitate the segmented cells. We save the results to the same result set. We do plan to improve this user interface so that this does not need to be typed in multiple times. We run the algorithm and in this case wait about 25 seconds. We again see that the algorithm completed successfully. We click to view the results and see that we now have quantitative values for over 1,800 cells, where each cell is a row in this table. Columns represent cell morphological features or biomarker intensities. Although there are a great many biomarkers in this example, Thrive can be used with single-channel fluorescence images up to very high-dimensional hyperplex fluorescence images, all with the same relative ease. These box plots automatically show the distribution of several interesting immune markers across the entire image. We can also create a rectangular region of interest to query the expression values at regions within the tumor. The box plots automatically update as we do so. This is like talking to the tissue. To query expression patterns at different regions, we can use this feature to begin to assess how biomarker expression changes spatially throughout the tumor. Next, we will show an example of a heterogeneity analysis algorithm that is currently implemented. We will calculate the quadratic entropy value, which is a measure of ecological diversity, for regions of interest or neighborhoods around each cell. Feature index 19 corresponds to mean expression of CD20 in an internal table of features. Future releases will extract biomarker names from the data table for ease of use. This calculation runs very quickly for all the cells. Again, we scroll down to see the list of algorithm executions. And we now have a new column of data called diversity, which in this case is quadratic entropy of CD20 in a neighborhood around each cell. The histogram shows the spread of diversity values within the region of interest. Again, the plots update automatically when the region of interest is moved across the tissue. We can select regions of interest to assess the changes in heterogeneity between different spatial components of the tumor sample. Going forward, we expect Thrive to be a powerful software workbench for experimenting with novel algorithms for spatial tumor heterogeneity. The trial release shown here contains a working algorithm for each of the steps of our analysis pipeline. The alpha version, currently in development, will include multiple heterogeneity algorithms and an SDK to easily add new ones. Beyond that, we will include capabilities for outcome analysis to begin determining which heterogeneity algorithms may be the most significant for predicting clinical outcome.